Hello everyone, welcome to Ishara Learnings. I'm really excited to have you here. Let me start with a simple question. Every day, millions of people take medicines, tablets, syrups, injections, trusting that they are completely safe. But have you ever wondered who ensures that these medicines are free from harmful microorganisms? Because even a tiny invisible microbe can contaminate a whole batch of life saving drugs. This is where pharmaceutical microbiology becomes extremely important. In this course, we will explore how microbiologists protect medicines, ensure quality, and safeguard patient health. So let's begin this fascinating journey into the world of pharmaceutical microbiology with Ishara Learnings. Today I am here with a demo class on pharmaceutical microbiology. Before we go deeper, let me give you a simple example. Imagine a pharmaceutical company produces an injectable medicine that will go directly into a patient's bloodstream. Now think about this. If that injection contains even one harmful microorganism, it can cause a serious infection and put the patient's life at risk. This is why microbiologists test every batch of medicines to make sure they are free from microbial contamination. Let me give you another example. When pharmaceutical companies manufacture syrups, tablets, or creams, microorganisms from air, water, equipment, or even human handling can contaminate the product. That is why pharmaceutical industries perform tests like sterility testing, microbial limit testing, and environmental monitoring. So, in today's lecture, we will understand how microbiology protects medicine quantity, quality, and patient safety. And if you want to build a career in pharmaceutical microbiology, this course is specially designed for you. So, let's begin our learning journey. Before we move forward in the syllabus, let's take a moment to understand something very important. Imagine you go to a pharmacy and buy a medicine for your headache or fever. You take that medicine with full trust that it is safe for your body. But have you ever thought about how pharmaceutical companies make sure that medicine is completely safe? The answer lies in pharmaceutical microbiology. Microbiology plays a very important role in the pharmaceutical industry. First, it helps in ensuring product safety. Every medicine must be tested to make sure it does not contain harmful microorganisms that could affect the patient. Second, microbiology helps in preventing microbial contamination during manufacturing. Even a small amount of contamination from the environment can affect the quality of a pharmaceutical product. Third, microbiologists help maintain quality and regulatory compliance. Pharmaceutical companies must follow strict quality standards to ensure that medicines are safe and effective. And finally, microbiological testing helps companies meet regulatory requirements before the medicines are released into the market. So, in simple words, microbiology acts like a safety guard that protects medicines from contamination. Without microbiology, producing safe and reliable medicines would not be possible. That is why pharmaceutical microbiology is such an important field in the pharmaceutical industry. Now, you might be wondering who exactly can join this course. The good news is that this course is designed for a wide range of learners. If you are a fresher who wants to start a career in the pharmaceutical industry, this course will help you understand the fundamentals of pharmaceutical microbiology. If you are a pharma or life science student, this course will strengthen your understanding of how microbiology is applied in real pharmaceutical manufacturing. This course is also useful for working professionals who want to improve their knowledge or move into microbiology related roles. If you are working in quality control or quality assurance, this course will help you better understand microbiological testing and compliance requirements. And even if you are simply interested in microbiology, you are absolutely welcome to join. The best part is that even beginners can follow this course because everything will be explained step by step in a simple and practical way. So, whether you are a student, a fresher, or a working professional, this course can help you build a strong foundation in pharmaceutical microbiology. Now that we understand the importance of pharmaceutical microbiology, let's take a quick look at what this course will cover. In this course, we will start with the fundamental theory concepts of pharmaceutical microbiology so that you build a strong foundation. But this course is not just about theory. We will also discuss practical industry knowledge that is actually used in pharmaceutical companies and microbiology laboratories. You will also learn about important regulatory guidelines that pharmaceutical companies must follow to ensure product safety and quality. And most importantly, throughout this course, I will share real time pharmaceutical examples so you can understand how these concepts are applied in the real pharmaceutical industry. By the end of this course, you will have a clear understanding of pharmaceutical microbiology from both theoretical and practical perspectives. So let's move forward and start exploring this fascinating subject step by step. Now let's begin with the introduction to pharmaceutical microbiology. Before we go deeper, let me ask you a simple question. Have you ever wondered why food spoils if it is left outside for a long time? Or why doctors prescribe antibiotics when we get bacterial infections? The answer lies in microorganisms. Pharmaceutical microbiology is a branch of biology that focuses on the study of these tiny living organisms, which are too small to be seen with our naked eyes. These microorganisms include bacteria, fungi, viruses, protozoa, and algae. Let's look at a few simple examples. For example, bacteria can sometimes cause infections, but they are also studied in pharmaceutical laboratories to understand contamination and drug safety. Fungi, such as molds, can grow on food or pharmaceutical products if proper environmental control is not maintained. Viruses are responsible for many diseases, which is why pharmaceutical companies develop vaccines and antiviral drugs to control them. Every organisms like protozoa and algae are studied in microbiology because they can affect water systems and environmental conditions used in pharmaceutical manufacturing. All these microorganisms are extremely small, which means they cannot be seen with the naked eye and can only be observed using a microscope. And understanding these tiny organisms is very important because they can directly affect the safety, quality, and effectiveness of medicines. So, in pharmaceutical microbiology, we study these organisms to ensure that medicines remain safe for the patients. Let's understand microbiology lab design with a simple real life example. Think about a hospital operation theater. Doctors follow a strict pathway before entering the operation room. They wash their hands, wear sterile gowns, masks, and gloves. They cannot directly walk from the straight into the surgery room. Why? Because even a small contamination could affect the patient. The same principle applies in a microbiology laboratory. A microbiology lab must be carefully designed to control the flow of people and materials so that contamination does not occur. First, let's talk about personal flow. Imagine a scientist coming from outside the building. Before entering the testing area, they pass through a gowning room where they wear lab coats, gloves, hair covers, and sometimes masks. This prevents microorganisms from the outside environment entering the laboratory and contaminating samples. Now let's look at material flow. Samples coming from manufacturing areas must enter the laboratory through a designated sample receiving area. After that, they move to the testing area and finally to waste disposal or sterilization areas. The movement of materials should always go in one direction to prevent contamination. Now let's understand an important concept called cross-contamination. Cross-contamination means microorganisms transferring from one sample, surface, or person to another. Let's take a simple example from the kitchen. If you cut raw chicken on a cutting board and then use the same knife and board to cut salad, vegetables without cleaning them, bacteria from the chicken can transfer to the vegetables. That is cross contamination. The same thing can happen in a microbiology lab. For example, if a microbiologist handles a contaminated sample and then touches another sterile sample without changing gloves, microorganisms can transfer to the sterile sample. This could give incorrect test results. Another example is person to sample contamination. If someone coughs, talks too closely to open culture plates, or touches samples with unsterile gloves, microorganisms from the person may contaminate the sample. There can also be sample to person contamination, where microorganisms from a contaminated sample spread to the laboratory worker. That is why microbiology labs are designed carefully to prevent contamination from person to sample, contamination from sample to person. A well-designed microbiology laboratory ensures accurate test results, safety for laboratory workers, and protection of pharmaceutical product quality. So remember, microbiology lab design is not just about equipment and instruments, it is about controlled movement, proper procedures, and preventing contamination at every step. Now let's talk about media preparation and growth promotion testing, which is one of the most important activities in a microbiology laboratory. Think of culture media as the food for microorganisms. Just like plants need soil, humans need food and nutrients to grow, microorganisms also need the right nutrients to grow in the laboratory. Let me give you a simple real life situation. Imagine a pharmaceutical company is testing a water sample from its manufacturing plant. The microbiologist collects the sample and wants to check if any bacteria are present, but bacteria cannot be seen directly. So the microbiologist places the sample in a culture medium which provide nutrients that allow microorganisms to grow and form visible colonies. Before using that media, the lab must make sure it is prepared correctly. For example, dehydrated media powder is received from the manufacturer and stored under proper conditions to maintain its quality. Then the microbiologist prepares the media by dissolving it in purified water according to the manufacturer's instructions. After that, the media is sterilized in an autoclave to remove any unwanted microorganisms. Now, here comes an important step, that is growth promotion testing. Let's say the microbiologist prepared a batch of agar plates. Before using these plates for testing pharmaceutical samples, the lab must confirm that the media is capable of supporting that microbial growth. So the microbiologist intentionally introduces a known microorganism into the media. For example, a known bacterial culture is inoculated onto the agar plate. If the microorganism grows properly, it confirms that media is suitable for microbiological testing. But if there is no growth, it means the media is not working properly and cannot be used for testing pharmaceutical samples. That is why Every lot of media used in pharmaceutical microbiology laboratories must undergo growth promotion testing to ensure the reliable results. Now let's understand the handling of cultures in a very simple way. In microbiology laboratories, we work with standard microbial cultures, which are known microorganisms used for testing and validation. To understand this better, let's use a simple example. Imagine you want to grow a plant. You first need a healthy seed. If the seed is good and you plant it in proper soil, the plant will grow properly. But if the seed is damaged or contaminated, the plant will not grow correctly. In microbiology, microbial cultures work just like the seeds. For example, when microbiologists want to check whether culture media can support microbial growth, they add a known microbial culture to the media. If the microorganism grows properly, it confirms that the media is suitable for testing. Let's take another example. Suppose a microbiologist is performing a microbial test for pharmaceutical water. To confirm that the test system is working properly, the lab uses a standard microbial culture as a control. This is similar to planting a seed to check whether the soil is capable of supporting that plant growth or not. But just like seeds must be handled carefully, microbial cultures must also be handled very carefully. If cultures are handled improperly, they can contaminate other samples and affect test results. That is why microbiologists handle cultures inside the laminar airflow cabinets and follow strict laboratory procedures. So, in simple words, microbial cultures act like seeds that help microbiologists confirm that laboratory tests are working correctly. Now, let's talk about pharmaceutical water systems, which are extremely important in the pharmaceutical industry. Water may look simple, but in pharmaceutical manufacturing, it is actually considered one of the most critical raw material. Let's understand this with a simple example. At home, we use tap water for washing dishes, cleaning floors, or watering plants. This water is fine for household use, but it still contains minerals, impurities, and microorganisms. Now imagine using that same tap water to produce an injectable medicine that will go directly into a patient's bloodstream. That would be very dangerous. That is why pharmaceutical companies use different types of water depending on the purpose. First is raw water. Raw water is like the water that comes from natural sources or municipal supply. It is the starting point, but it must go through several purification steps before being used in the pharmaceutical processes. Next is the purified water. Think of purified water like filtered drinking water at home, but much cleaner. This type of water is commonly used in pharmaceutical plants for cleaning equipment, preparing solutions, and laboratory work. Now let's look at water for injection, also known as WFI. This is one of the purest forms of water used in the pharmaceutical industry. For example, when producing vaccines or injectable medicines, the water used must be extremely pure because it will enter the human body directly. Even the smallest contamination could affect patient safety. Another type is pure steam. Pure steam is used for sterilization. For example, when laboratories sterilize equipment in an autoclave, steam helps destroy that microorganisms and maintain sterile conditions. So in pharmaceutical manufacturing, water is not just water. It must be carefully generated, purified, and monitored to ensure it meets strict pharmaceutical standards. That is why pharmaceutical water systems are constantly validated and tested to ensure the safety and quality of medicines. Think about making tea at home. If the water is dirty, the tea will also be contaminated. In the same way, if pharmaceutical water is contaminated, the medicine produced using that water can also become unsafe. Now imagine this. A pharmaceutical company produces thousands of tablets every day. Patients trust that those medicines are safe. But have you ever thought, where does the water used in pharmaceutical manufacturing come from? And how do we make sure it is safe? In pharmaceutical industries, water is not just ordinary water. It is used in drug manufacturing, equipment cleaning, and even injections. Now think about a simple example. If you fill a clean bottle with water and leave it open for a few days, tiny microorganisms from the air can enter and start growing. Even though the water may look clear, microbes may already be present. This is exactly why water sampling and testing is extremely important in pharmaceutical industries. In this chapter, we will learn how water samples are collected from the system, what testing methods are used to detect the contamination, how data is recorded properly, and how monthly and annual trending helps us to monitor the health of the water system. For example, if microbial counts slowly increase over several months, trending helps us detect the problem early before it affects our product quality. So remember, water monitoring is not just testing, it is a system that ensures consistency, safety, and patient protection. Let's explore how pharmaceutical industries perform water sampling and testing step by step. Now, let me ask you a simple question. Imagine you cleaned your room in the morning. Everything looks perfectly clean. But if you leave the windows open, dust and microorganisms from the air will slowly enter again, right? The same thing happens in pharmaceutical clean rooms. Even though the room looks perfectly clean, microorganisms can still be present in the air, on surfaces, or even carried by people working inside the room. That is why pharmaceutical industries perform something called environmental monitoring. Environmental monitoring helps us check whether the clean room environment is truly clean and controlled. There are three common methods used. First is active air sampling. In this method, special instruments actively pull air from the clean room and collect microorganisms on agar plates for testing. Second is passive monitoring, also known as settle plates. Here, agar plates are simply exposed to the environment and microorganisms from the air naturally settle onto that surface. And the third method is surface monitoring. This checks microorganisms present on surfaces like tables, equipment, walls, or gloves using the contact plates or swabs. Now, here is the most important part. All this data is analyzed over time using trend analysis. This helps pharmaceutical companies set alert limits and action limits to detect contamination early and maintain cleanroom control. So remember, environmental monitoring is not just testing the environment, it is a system that protects product quality and the patient safety. In the next section, we will explore these monitoring methods in more detail. Let's imagine something very simple. Suppose you buy a bottle of juice from a store. The juice looks clear and fresh, but one important question still remains. Is it really safe to consume? The same question applies to pharmaceutical products. Even if a medicine looks perfectly clean, microorganisms may still be present in small numbers. That is why pharmaceutical laboratories perform something called the microbial limit test. The microbial limit test helps us determine how many microorganisms are present in a product such as water, APIs, or finished pharmaceutical formulations. But in some cases, we also need to check for specific harmful microorganisms. For example, E. coli, which is commonly associated with the contamination from the improper hygiene, Salmonella, which can cause serious infections, Staphylococcus aureus, often found on human skin, Pseudomonas aeruginosa, which can survive in the water systems. Detecting these microorganisms is extremely important to ensure the safety and quality of pharmaceutical products. Now, there is another important test called the bacterial endotoxin test. Even when bacteria are no longer alive, they can release substances called endotoxins. These endotoxins can cause fever or serious reactions in patients, especially in injectable drugs. Therefore, pharmaceutical companies perform endotoxin testing used to make sure that the product is safe for the human use. So remember, microbial testing is not just about finding microorganisms, it is about protecting the patient's safety and maintaining the product quality. In the next section, we will explore how these tests are performed in the laboratory. Let's imagine a real situation. A patient in a hospital receives an injectable medicine. This medicine goes directly into the bloodstream. Now think about this question. What would happen if that medicine was contaminated with microorganisms? It could cause serious infections or even life-threatening complications. That is why pharmaceutical industries perform sterility testing. Sterility testing ensures that sterile products such as injections, eye drops, and IV fluids contain absolutely no microorganisms. Now let's talk about another concept, that is stability testing. Imagine you buy a medicine today, but you may use it several months later. The company must make sure that the medicine remains safe, effective, and stable throughout its shelf life. This is why pharmaceutical laboratories perform stability testing under different temperature and the storage conditions. Next, we have bioassays. Bioassays are used to determine the biological activity or potency of certain drugs, especially antibiotics and vitamins. There are two commonly used methods. The first is the cup plate method, where antibiotics diffuse through agar and create a clear zone that shows their effectiveness against microorganisms. The second is the turbidimetric method, where microbial growth in liquid media is measured to determine the drug potency. So remember, sterility testing protects patients, stability testing ensures product quality over time, and bioassays confirm that medicines work as intended. Together, these tests play a critical role in pharmaceutical quality control and the patient's safety. Let's start with a simple example again from our daily life. Imagine you open a bottle of syrup or eye drops. After opening, you may use that product for several days or even weeks. During this time, the product can be exposed to air, hands, or the environment. So, the important question is, how do we make sure microorganisms do not grow inside that product? This is where preservatives play a very important role. Preservatives are special substances added to the pharmaceutical formulations to prevent the growth of microorganisms and protect the product during its shelf life. But simply adding a preservative is not enough. Pharmaceutical companies must confirm that the preservative is actually effective. This is done through preservative efficacy testing. In this test, scientists intentionally introduce known microorganisms into the product and then observe whether the preservative can successfully control or eliminate microbial growth over time. Another important concept we will discuss is analytical method validation in microbiology. Method validation ensures that the testing methods used in the laboratory are accurate, reliable, and reproducible. Finally, we also perform recovery studies. where known microorganisms are introduced into the samples to confirm that the testing method can correctly detect and recover them. So remember, preservative efficacy testing protects the product from contamination, and method validation ensures that our laboratory testing methods are trustworthy and scientifically reliable. Together, these steps help maintain pharmaceutical product quality and patient safety. Let's imagine a simple situation. Suppose the doctor is about to give a patient an injection. That medicine goes directly into the bloodstream. Now think for a moment. What would happen if even a tiny microorganism was present in that injection? It could cause a serious infection. That is why in pharmaceutical industries we must make sure that all equipment, materials and products are completely sterile. One common method used is moist heat sterilization, which is performed using an autoclave. You can think of an autoclave like a very powerful pressure cooker used in laboratories. Just like a pressure cooker uses steam and pressure to cook food quickly, an autoclave uses steam and high pressure to destroy microorganisms and their spores. Another method is dry heat sterilization. For example, glassware such as petri dishes, pipettes, and metal instruments can be sterilized using hot air ovens, where very high temperature kill microorganisms. Sometimes heat cannot be used. For example, vaccines, antibiotics, or heat sensitive solutions may be damaged by high temperatures. In such cases, scientists use filtration methods where the liquid passes through very fine filters that trap microorganisms. But how do we confirm that sterilization was successful? For this purpose, we use biological indicators. These indicators contain highly resistant bacterial spores. If the sterilization process can kill these spores, we can be confident that the process was effective. Finally, pharmaceutical facilities also use disinfectants to clean surfaces like floors, walls, equipments, and workbenches. These disinfectants are carefully validated using special testing methods to ensure that they can effectively control microbial contamination. So remember, sterilization, biological indicators, and disinfectants work together to maintain a safe and contamination-free pharmaceutical environment. Let's imagine a real hospital situation. A nurse is about to give a patient an injection or IV medicine. This medicine goes directly into the bloodstream. Unlike tablets, there is no barrier in the body to stop microorganisms. So even one tiny bacterium in that injection could cause a serious infection. That is why pharmaceutical companies use something called aseptic processing. Aseptic processing means manufacturing sterile medicines in a completely controlled clean room environment where every step is carefully designed to prevent contamination. For example, operators wear special sterile gowns, gloves, masks, and head covers, and the filling process is performed under laminar airflow to keep microorganisms away. But here is an interesting question. How do we know that the aseptic process is actually working properly? This is where media field studies, also called process simulation studies, come into the picture. Instead of filling the vials with real medicine, scientists fill them with sterile nutrient media, a liquid that microorganisms love to grow in. Now imagine this. If even one microorganism accidentally enters the vial during filling, the nutrient media will allow it to grow and the vial will become cloudy or show microbial growth after incubation. In this way, scientists can confirm whether the aseptic manufacturing process is truly sterile. Sometimes, if microbial growth is detected, it is called a media fill failure, and the company must investigate the cause, such as operator error, equipment issues, or environmental contamination. So remember, aseptic processing protects sterile products, and media field studies help confirm that the entire manufacturing process is working safely and effectively. This is one of the most critical steps in sterile pharmaceutical manufacturing. Now let's imagine a real laboratory situation. A microbiologist performs a test on a pharmaceutical product and the result suddenly shows very high microbial growth, even though the product is expected to be sterile. This type of unexpected result is called OOS, which stands for out of specification. For example, if the acceptable microbial limit is not more than 100 CFU, but the test result shows 300 CFU, this result becomes an OOS result. Whenever out of specification occurs, the laboratory must carefully investigate the root cause. The contamination could come from improper sampling, environmental contamination, equipment issues, or human error. Now let's talk about the cleaning validation. Imagine you bake a cake in a pan and later you want to cook a completely different dish in the same pan. If the pan is not cleaned properly, some ingredients from the previous recipe may remain and affect the new dish. The same concept applies in the pharmaceutical manufacturing. After producing one product, all equipment must be cleaned thoroughly so that no residues, microorganisms, or chemicals remain before manufacturing the next product. To confirm this, scientists perform cleaning validation using swab methods and rinse methods. For example, a swab is used to collect residues from equipment surfaces and then the sample is tested to check if any contamination remains. Finally, we have the waste handling. In microbiology laboratories, biological waste such as used culture plates, contaminated media, and microbial samples cannot be thrown away like normal garbage. Instead, they must be properly sterilized, treated, and disposed of safely to protect laboratory workers, the environment, and the public health. So remember, handling out of specification results ensures product quality, cleaning validation prevents cross contamination, and proper waste handling maintains laboratory safety and environmental protection. All these steps are essential parts of pharmaceutical microbiology and GMP practices. Next is the identification, guidelines, and audits. Let's imagine a real situation in a pharmaceutical microbiology lab. During routine environmental monitoring, a scientist notices unexpected bacterial colonies growing on a culture plate. Now, the big question is what microorganism is this? Is it a harmless environmental bacterium or a dangerous contaminant like E. coli or Pseudomonas that could affect product safety? This is where microbial identification becomes very important. For example, scientists may use gram staining, biochemical tests, or automated systems to identify the microorganism, just like a detective identifying a suspect using fingerprints. Think of it like crime investigation. When police find evidence at a crime scene, they analyze fingerprints and DNA to identify the person responsible. In microbiology, we analyze microbial characteristics to identify the microorganism responsible for the contamination. Now let's talk about international guidelines. Organizations such as USP, EP, and WHO create standards that pharmaceutical companies must follow worldwide. For example, if a medicine is manufactured in India, Canada, or Europe, these guidelines ensure that the product meets the same quality and safety standards everywhere. Next comes data integrity, which is extremely important in pharmaceutical laboratories. Imagine if a scientist performed a test but later changed the result to make the product look acceptable. That could be very dangerous. That is why pharmaceutical companies follow the ALCOA principles, ensuring that all laboratory data is attributable, legible, contemporaneous, original, and accurate. Finally, we have the audits. Think of an audit like a health inspection in a restaurant. When health inspectors visit a restaurant, they check whether the kitchen is clean, food is stored properly, and safety rules are followed. Similarly, during a pharmaceutical audit, regulatory authorities check laboratory records, equipment calibration, training records, and testing procedures to ensure the company follows good manufacturing practices. So remember, microbial identification helps detect contamination, guidelines ensure global quality standards, data integrity protects reliable results, and audits confirm that everything is done correctly. All these steps are essential to ensure safe medicines for patients around the world. Before we end this session, think about something important. Every medicine that reaches a hospital, pharmacy, or patient has passed through strict microbiological testing and quality control. Behind the safety of those medicines are skilled pharmaceutical microbiologists who ensure that every product is free from harmful microorganisms. In this demo lecture, you have only seen a small window into the world of pharmaceutical microbiology, but the real journey is much bigger. In the complete Ishara Learnings course, we will go step by step through the practical concepts used in pharmaceutical industries, from environmental monitoring and sterility testing to aseptic processing, cleaning validation, audits, and regulatory guidelines. You will not only understand the theory but also gain the industry oriented knowledge that companies expect from microbiology professionals. Many students struggle to understand how pharmaceutical microbiology actually works in real laboratories. This course is designed to make those concepts simple, practical, and easy to understand. So, if you want to build confidence and prepare yourself for a career in pharmaceutical company or in a pharmaceutical microbiology or quality control laboratories, the full course will guide you through that journey. Thank you for watching this demo session. I look forward to seeing you in the complete course at Ishara Learnings, where we will continue learning and exploring this fascinating field together. Until then, stay curious, keep learning, and keep moving forward in your scientific journey.